So welcome everyone today, and thank you for joining us for the Kirby Institute seminar series. My name is Rebecca Guy and I'm Head of the Surveillance, Evaluation and Research Program at the Kirby Institute. Before we start, importantly and sincerely, I'd like to show my respects and acknowledge the Gadigal people who are the traditional custodians of the land where the Kirby Institute is based, and pay my respects to elders past and present, and extend that respect to Aboriginal and Torres Strait Islander people who've joined the seminar today. So in true keeping with the seminar series, just a few housekeeping notes. As you probably have realised, because you've joined virtually, this format is virtual only. And we will start off with a 40 to 45 minute presentation, followed by a 15-minute Q&A at the end. However, I would encourage you to ask your questions throughout the talk. Just helps us in the end try and see what the priority questions are and use that 15-minute wisely. Now I'd like to introduce our speaker, the illustrious Professor Deborah Williamson. And like a true Australian, I refer to Deborah as Deb. Many of you know Deb, but she's a very well-known, respected and admired researcher in Australia and internationally. And we are fortunate she's able to give this talk today. As a bit more background, Deb is a clinician-scientist with leadership roles in clinical and public health microbiology and virology. She's the director of the Victorian Infectious Disease Reference Laboratory, otherwise known as VIDRL, and Professor of Public Health Microbiology at the Doherty Institute, University of Melbourne. For me personally, I've known Deb for more than 5 years and I've had the great privilege of collaborating with Deb on a range of projects related to antimicrobial resistance, diagnostic development and evaluation, the COVID-19 point-of-care testing program and the response, and the recent POC23 conference. I've also admired her game-changing research, the development and integration of genotypic methods into public health surveillance. So without further ado, I'm very excited to hear Deb's talk today, and I'm sure you are too, so I'll hand over to Deb to give a presentation. Thank you.
Thanks, Beck, for that really nice introduction. And I'd also like to acknowledge the Wurundjeri people, who are the traditional custodians of the land where I am today in Parkville, and I pay my respects to their elders past and present. And I feel really honoured to be giving this talk to the Kirby today. Beck has been is a wonderful collaborator of mine, but more broadly, I really admire the work that comes out of the Kirby. I think in the POC23 conference was a really, pivotal moment for many. I think just highlighting, the leadership that Kirby has shown in this space. So what I want to do today is just, to talk more broadly about how we have applied innovations in the area of laboratory science, specifically to the detection and control of infectious diseases. And I guess I'm going to focus not on a particular pathogen or topic, but on three main areas to give a broad overview of how laboratory science can innovate in pandemic and outbreak and even peacetime settings. So I'll be talking about COVID-19 testing. I'll be talking about how we've applied genomics to STI tracking. And then I'll finish by talking a little bit about what I think are some innovations in the point of care testing space. Before I start, actually, I want to do acknowledgements up front because I think it's really important for me to highlight that I'm presenting this work on behalf of a huge team of people, both in my lab with our wonderful collaborators at Melbourne Sexual Health Centre, at VIDRL, and many others, both nationally and internationally. So this is a huge team effort. And I'll be presenting some work as well from some PhD students in my lab. All right, but I want to start by talking about some of the diagnostic innovations that happened during the pandemic. I think one of the most critical parts in laboratory science during the pandemic was right at the start, when the virus was isolated. And I'm sure that you've heard many times, but VIDRL, who, then under the leadership of Mike Catton, was the first lab outside of China to grow but most importantly, share the virus. And that isolate VICO1 still pops up in many publications. And I think it was it really catalyzed a lot of diagnostic development, not just in Australia, certainly not just in Victoria or Australia, but globally as well. But again, and it's, sometimes it's easy to forget how challenging those early days of the pandemic were. But I think, many of us, and including many people I'm sure on this call, knew early on that this would involve laboratory testing on a scale that we'd never seen before. It became clear to us that the scale of the testing response would be enormous, but importantly, the hardware, the reagents, the people needed to bring about that testing response would be unprecedented. And very early on, we found ourselves with significant supply chain shortages, supply of PPE, supply of swabs, supply of laboratory reagents. So at that point, we thought, okay, how can we try and mitigate this? And one of the first things that we did was actually work with industry to try and print our own swabs. And I do recall, in the early days, a few of us were driving around in our cars between different labs in Melbourne, looking for, trying to move swabs between places. I mean, it got that bad. So we worked with a company and we came up with a prototype swab. This was the final version. We called it the pineapple. And actually that has got, throughout the 2020, that went through the TGA registration process and is now available as a swab. But that was, for us, that was the first time that we thought, okay, we really need to think outside the box. And importantly, we need to think outside our traditional working partners, and actually seriously start to work with industry to help in the pandemic. The other way that we tried to mitigate not only the swab shortage, but the PPE shortage was to try and think of different ways of sample collection. And this was one of the very first papers that came out looking at saliva as a specimen for SARS-CoV-2 detection. And essentially what we showed was that collecting saliva was no worse than using a nasal, it was not much better, but it was no worse than the yield from a nasopharyngeal swab. And what that meant was, I guess, that we were able to use that initial pilot as a justification for a broader rollout. And I know that this caused challenges across the country, because certainly in Victoria, for obvious reasons, we had to, I think, scale up and innovate our testing response in the latter stages of 2020, whereas, potentially other jurisdictions were not in that position. I'll get to that later on in the talk. But this was really kind of taken on by the state government and expanded big time as a way of trying to facilitate access to testing. And one thing that we did, and I do see this as a sort of central role as a reference laboratory, is to try and decentralize testing where possible. And so one of the things that we did was to develop panels of saliva samples and distribute them to laboratories throughout Victoria. And we moved on from the initial dribbling method of collecting saliva to a salivary swab, and sent out samples to different labs. They were able to validate that themselves and then scale up that salivary testing response across the state. Where was that used? Well, two main settings. One, it was used in the hotel quarantine program, and I will get to that. But it was also used in healthcare worker surveillance. And I don't think that kind of daily or high frequency swabbing would have been possible without that more acceptable salivary swab method. So again, an example of how a small innovation can be scaled up. I think it's about 700,000 salivary swabs have been taken now. All right, I'm going to move on to another innovation, which Beck mentioned in her introduction around genomic testing, and this did exist before before COVID, genomic sequencing, but it was still kind of entrenched a bit in research. But the COVID pandemic really brought, I think, genomic sequencing to the forefront. This is a graph of all of the cases in Victoria. And again, if a graph could tell a million words, this would be one. And this small, it looks like a small molehill here compared to this Omicron mountain, but this period here, really dark days, I think, during the pandemic, this initial phase, the national lockdown, and then the Victorian lockdown here. And this story can also be told through genomics. And so this was some early work that we did with the genomics data. This is actually just showing the number of cases prior to and after that first national lockdown. So increasing number of cases, mostly from overseas, and then the national restrictions having a very quick impact on the number of cases. Obviously, that closure of the international border being key. This is a story in genomics of the early days of 2020. So each line here, and this will be a common theme throughout the talk, these horizontal lines representing a genomic cluster. So cases that are linked through genomic sequencing. And essentially what you can see here, multiple importations, short chains of transmission shut down. One here going through the northern suburbs of Melbourne, but again, shut down here, but mostly international importation, short chains of transmission shut down. And again, you can see that following the restrictions, the reproductive number of the virus came down from over 1 to less than 1 here. So that's the first part of the COVID pandemic in genomics. And then obviously across the country, this hotel quarantine program was stood up. And again, it's easy to forget that this was a massive exercise, setting up this program. The logistics involved, I think, were mind-boggling. Selecting the hotels, insurance, private security, transfers, cleaning, catering, a huge program set up within a very, very short time period. Again, obviously some challenges with that. So these were the first cases. I'll just go back here. These were the first cases that I showed here. Initially, national public health restrictions, and again, these are showing cases in Victoria, but clearly something bubbling along here, and then this second wave here in Victoria. And again, this story can be told using genomics. And so this is the first graph that I showed, these multiple international importations shut down. This is the period here where hotel quarantine had been introduced. And this period here is where clearly there was something going on in Victoria, and there was the second lockdown here. Now, this is really the key part. What we can see here, several breaches of the hotel quarantine process, but one in particular leading to that massive second wave here. Of note, the salivary testing program that I mentioned before was introduced around here, and I like to think of that program as a sort of the fragile shield, if you like, not the only shield, but part of a shield around the hotel quarantine program subsequent to that breach and second lockdown. But if ever there was a picture, I think, that told the story of 2020 in Victoria in genomics, this would be it. And obviously that led to a very, very long demoralizing lockdown with ongoing societal and ethical ramifications. And of course the hotel quarantine inquiry, which I think was one of the first inquiries to use genomic sequencing as part of the findings, and not just in Australia but globally. One thing I think that many of us felt a little bit uncomfortable about was, the daily press conferences mentioning genomic sequencing, and it got to a part where it became, I think, a bit contentious about even individual identification, and there's no, and still is no clear framework around how do we report pathogen genomic results back to individuals, or indeed whether we do that. And I think we're very good now at reporting these results in a research context, and also back to public health units, but not really back to the individuals. And so this is an area where, several of us, I guess, are actively thinking about, and how do we balance public health with individual autonomy and privacy? And we've sort of called that pathogen genetic counselling. This is an area I think that appropriately will expand markedly over the next 5 to 10 years, and should. All right, I'm going to finish the COVID part of the talk by talking about rapid antigen tests. And I know a number of people at the Kirby, including Beck, were heavily involved in, rolling these out across the country. But again, it's easy to forget that Australia was a very late adopter of rapid antigen tests, particularly for self-testing. November 2021 was actually when they were rolled out, and which was useful in advance of that huge Omicron wave. But there was a lot of, I would say, discussion about their utility and the fact that they were less sensitive than PCR. Different schools of thought, any test is better than no test, or we shouldn't be using them, they're not perfect. So it was a very interesting and challenging time around the adoption of rapid antigen tests in Australia. And one thing that we had to do early on was again use these in a research context to show public health utility before they were rolled out at scale. And this is one study that we did in Melbourne, where in the Royal Melbourne Hospital Emergency Department, I have to say they were fantastic to work with during the pandemic, very open to trialing different diagnostic methods, including the saliva testing, including the 3D printed swabs. And for me, that was a valuable lesson in, really having those deep, enduring clinical collaborations where you can translate laboratory innovations into clinical practice quickly. So this is what we did with rapid antigen tests. We took rapid antigen tests from people who had suspected COVID at triage, and they had a concurrent rapid antigen test and a PCR test as well. And then we simply compared the sensitivity of rapid antigen tests with PCR. And we did not find anything different from the rest of the world, so we knew that they were less sensitive. But what was interesting was, in the context of a high prevalence at that time, they had a pretty good positive predictive value and negative predictive value as well. And what we found that was most useful, and I think really led to their adoption, particularly at Royal Melbourne, was that we were able to show a reduced length of stay in the emergency department when somebody was had a positive RAT test, and that they were able to go up quickly to a COVID ward. So they weren't hanging around waiting for the result of the gene expert, etc. They were just triaged right up there. I think these kind of, I would have liked to have seen these kind of studies done multi-site across the country. And again, I'll get to this later in the talk, but I don't think that we were as good as we could or should have been in joining up these kind of studies across the country. One thing that we were asked to do in my lab was to undertake post-market surveillance of these tests by the TGA. And this was a huge and challenging piece of work, I have to say, but important, I think, because there hadn't been much experience with these kind of tests before. There'd been a little bit for flu, a little bit for RSV and some other diseases, but not at this scale. And so what we were able to do was to do a post-market evaluation of all of the tests now that came on the Australian market. All of those results and the protocol that we used are available on the TGA website. But I hope anyway that we were able to contribute to making sure that only good quality products came onto the market here. But what we had to do was, every time a new variant and I think certainly had good activity, or were able, I should say, to be picked up by the major kits on the market. But again, this was a challenge, and it involved a lot of work in the laboratory. All right, I'm going to move now to talk a little bit about some of the genomic work we've done on STIs. And again, this is, I've talked about how things that start in a research domain can be moved into public health. And this is something, it's not quite there yet, but I think it's within touching distance. This group, I think, who are on the who are listening at the moment, need absolutely no introduction to the importance of STIs as a public health problem, and will be more familiar than probably any group in the country with the multifactorial factors that contribute to the emergence and spread of STIs. We've used genomics for a couple of STIs previously, and a number of years ago now. So we used genomics to look at the emergence and spread of multi-resistant Shigella, which is still a problem, and to really show that there was, I guess, a major outbreak amongst MSM, which is still ongoing actually, and is a major public health problem. I'm not sure that we have adequately addressed the problem of sexually transmitted enteric infections, because it's something that I guess from a public health perspective, there's not entirely clear where it sits. Does it sit with the foodborne disease people? Does it sit with the STI people? I guess it would be great to see a joined up approach again across the country on how to best deal with sexually transmitted enteric infections. But anyway, what we did here was to show that with Shigella sonnei anyway, most of the cases of multi-drug resistant Shigella were imported, but what we're seeing in Victoria was this large outbreak amongst MSM. And that was the first time it had been, I guess, assessed epidemiologically, but this was the first kind of definitive genomic evidence for that. What this enables is, I guess, a bit more focused contact tracing and public health interventions. Similarly, we use genomics to look at gonorrhea. Again, these horizontal lines are genomic clusters. And what we're able to show here is that clusters of gonorrhea persisted over long periods of time, up to, and indeed probably beyond a year, which again was the first time I think that anybody had shown that genomically. And again, you can think, okay, well, along that kind of those chains of transmission, there might be multiple opportunities for public health intervention. And what we, again, were able to do, and this is, I guess, the genomic data in and of itself is not helpful unless you can link it up to epidemiological data. That's where the public health utility comes. And so we were able to link the gonorrhea genomic data with some epidemiological data to show for the first time that there was bridging of different gonorrhea lineages across different sexual networks. So across networks that predominantly included MSM, across heterosexual networks. Again, what that enables is tailoring, I think, of public health intervention. So there's no one size fits all to the problem of gonorrhea, but you can use genomics to look at emerging clusters or AMR clusters, or clusters in one particular geographic location. And again, this hasn't been widely adopted in real time, but the potential is absolutely there. So I guess more recently, we've been interested in whether, noting the significant public health challenges with syphilis, we've been interested to know whether we can apply this to the syphilis epidemic in Australia. Well, this has real inherent challenges, mainly due to the fact that Treponema pallidum is a nightmare to work with in the lab. It doesn't grow on an agar plate like Neisseria gonorrhea or Shigella, and it has a very small genome, and there simply is not much experience of looking at it genomically. There's a lot of experience in people working with Shigella or gono, but not so much with syphilis. So we needed to innovate, and we needed to develop a different way of undertaking genomic sequencing of Shigella. So we couldn't just grow it up on an agar plate, take the colony and sequence it as we would do for Shigella or gonorrhea. We needed a different approach. And the questions that we wanted to answer with this, specifically the public health questions, are, is a current syphilis outbreak in Australia is it, because we often hear the syphilis outbreak in Australia, but is it just the outbreak? Is it one strain of Treponema pallidum across the country, or are there multiple co-circulating lineages? Importantly, are they linked to globally circulating strains? So is there a distinct problem here, or a distinct kind of hypervirulent or hypertransmissible strain here? Or is it just, that we are part of a broader global community? And importantly, can we use genomics to define transmission networks of syphilis? So we used a different approach to sequence Treponema pallidum. I should just say that the other challenge with Treponema pallidum is that it's present at really low levels in clinical samples. So the way that we tackled this was to use something called a target enrichment approach. We used RNA baits with little bits of complementary baits to parts of the Treponema pallidum genome. And those baits were biotin labeled, and we hybridized those with, I guess, clinical samples. And we used the baits to pull out the Treponema pallidum genome using a magnetic bead capture approach, and then sequenced it. And we were able to recover whole genomes of Treponema pallidum using that approach. And I should just thank our collaborators at the Sanger Institute who kind of pioneered this approach, and we worked with them to develop this Treponema pallidum approach as well. And we were really pleased actually with how well it worked. And essentially what we showed was, and this was work that was led by a PhD student in my lab, fantastic student, Mona Tauk, and she really did a wonderful job with this. But what we showed was that the current syphilis epidemic in Australia, it's not one strain. So there were at least 4 different major lineages co-circulating in Australia. There are 4 major lineages co-circulating in Australia at the moment. So not just one distinct outbreak. 1, 2, 3, 4 here. 2 of them belong to the Nichols lineage, which is the sort of archetypal, if you like, Treponema pallidum lineage, and 2 of them belong to this newer SS14 lineage. What we were also able to do through something called a Bayesian reconstruction is to show that the major subgroups, these 4 major subgroups, have emerged in Australia over the last 30 years, so in the sort of latter part of the 20th century, and that they are closely related to globally circulating lineages as well. Again, we linked it up, excuse me, with genomic epidemiological data, and we were able to show that, certain, and again, these different, I guess, horizontal lines here represent different lineages, and we were able to show that different lineages of Treponema pallidum circulate across different networks. Having said all of that, unlike gonorrhea and Shigella, I have to say that I'm not convinced that that genomics, and this might sound like heresy for me because I love genomics, but I'm not convinced that genomics for syphilis tracking is something that can be used in real time, largely because A, it's a very laborious process to do, but also B, because Treponema pallidum mutates so slowly that the information that you get is not particularly useful in real time. Is potentially every kind of maybe a 5-year snapshot, but not so much in real time. Having said that, these data do provide the first genomic insights into the current syphilis epidemic here. But genomics is not, as I said, not something that should be a front and center of the public health response. As I'm sure many people here are aware, the absolute priority for syphilis control is improving access to testing and treatment. But I think these data go some way to highlight the fact that there are these multiple concurrent epidemics of syphilis. So a one-size-fits-all public health approach is not, may not be provide the best bang for buck. All right, I'm going to finish by talking about some, I guess, novel approaches to point-of-care testing here. And again, I just want to acknowledge the work that the Kirby has done in the point of care testing space. Not only is it from a research perspective always interesting and high quality, but from a public health perspective, I genuinely believe that has been transformative particularly for regional and remote Australia. I'm going to talk a little bit about how we've used CRISPR-Cas diagnostics specifically for the recent Mpox outbreak. So for those of you who are not familiar with the CRISPR-Cas system, it was originally identified as a bacterial immune system, but it's been used as a really versatile tool for gene editing. And now it's got significant applications in various fields, including diagnostics. And the basic principle behind using it for diagnostic involves this guide RNA recognizing a target sequence, whether it's a DNA or RNA sequence of interest. When the guide RNA binds to its target sequence, it triggers the activation of a Cas enzyme, in this case Cas12a, leading to this molecular response that can be easily detected and quantified. That's in very basic lay terms. But essentially, the sensitivity comes, or specificity, I should say, comes from having this guide RNA binding to the target sequence. So we applied this to the Mpox outbreak, and again, the Mpox, as we were kind of recovering in the lab from Omicron, the emergence of Japanese encephalitis. This happened last year. So last year was pretty busy. And the first cases were confirmed in May last year in the United Kingdom with patients who had no link to travel in an endemic country. And obviously it spread globally very quickly. And as of 15th of May, there's been over 86,000 cases in over 113 countries. Really significant outbreak last year, and a stark reminder, I guess, of how these things can kind of appear out of nowhere. So at the time, we've been working on CRISPR-Cas diagnostics for other STIs. And so what we were keen to do is to see whether we could apply that to that approach to the monkeypox virus. And essentially what we did was to take samples, extract DNA. We used an isothermal amplification approach. So this is amplifying up bits of the target sequence, but not using PCR. And I'll get to why that's important later. That should read 37, sorry. And then apply the CRISPR-Cas system to this and look at the readout on two things. One, a fluorescent readout in the laboratory, but second, a lateral flow readout. And this is where the point of care aspect comes in, because if you can get something onto a lateral flow device with a small point of care machine attached to it, then that makes it transportable, and it genuinely makes it point of care. What we're able to show with the fluorescent readout was that we got very good sensitivity, actually, and good specificity across a panel of other viruses that might cause skin lesions. And again, what we were very pleased to see is that we retained that sensitivity and specificity with a lateral flow readout. What we've been trying to do is to get that into a small point of care testing device. Why is this important? Well, having that isothermal amplification means that you don't need heat blocks, and that again makes it more transportable in settings where an electrical supply, etc., might be limited. And we applied it to a large panel of clinical samples that had come through Vidrol and got very good sensitivity and specificity. All right, I'm just going to come back and finish by trying to bring it all together by talking about how we have combined point of care testing with genomics. And again, I'm coming back to this graph because here, as I mentioned, self-testing took off in a big way. in Australia. And what that meant was that PCR testing essentially bottomed out. And I think the ratio of of antigen testing to PCR testing at the end of last year was about 3-to-1. So more rapid antigen tests than PCR tests. What that meant for genomic surveillance, which is traditionally done from residual PCR samples, is that there was a potential for a large kind of black hole in genomic surveillance. So what we wanted to do was to see whether we could continue to do genomic surveillance for SARS-CoV-2. I look at different variants, etc., from rapid antigen test devices. And so we took positive rapid antigen test devices and cracked them open. It was not a sophisticated approach. It literally was a palette knife from Bunnings. Extracted nucleic acid from the membrane, and then did PCR testing to see whether we could detect SARS-CoV-2, and then genomic, and then sequencing. We looked at two things. We looked at residual PCR diagnostic samples and put them on a rat test of samples that had come through and that we'd stored. But importantly, we also collected used rapid antigen tests, again, from our fantastic collaborators at Royal Melbourne ED, who saved them for us, and we picked them up. And what we're able to show is, yes, we were able to recover full-length genomes from positive rapid antigen tests. Unsurprisingly, this is a CT value using PCR, and as there were going along here, this axis here means that there's less virus, and unsurprisingly, the more virus there is, the more likely we were able to get a complete genome, and the depth was better as well. What's important here is that we didn't pick up these samples every day, and sometimes there was like a delay of several days before we picked up the samples, but we were able to recover full-length genomes for up to 8 days, actually, post collection. Certainly not in every instance, but we were able to do that, which, again, if you think about things like potentially doing large-scale surveys where people are able to post in their rapid antigen tests, or in remote settings where they may not be able to have ready access, I guess, to a sequencing or reference laboratory. And I'll just finish by presenting, I guess, some recent work that we've done looking at negative COVID rapid antigen tests. So somebody with respiratory symptoms, and there's a lot going around at the moment, and clearly we had a big flu and RSV season last year. So if somebody did a SARS-CoV-2 rapid antigen test and they had respiratory symptoms, but they didn't have COVID, or it was not positive for SARS-CoV-2, what else do they have? So what we did was to take a panel of other respiratory viruses that were, I guess, stored samples in vitro from patients with respiratory symptoms. We applied them to 2 different rapid antigen test devices, extracted nucleic acid, and looked to see whether, first of all, we could detect the same virus with PCR, and we saw a bit of a drop-off, but not that much, actually. And then secondly, whether we could recover viral genomes. And we used a metagenomic approach for this. And we were able to recover full-length genomes for influenza, adenovirus, metapneumovirus, RSV, and for influenza A, we were able to type those viruses. Why is that important? Well, I mean, pandemics will always happen. And in fact, there's a large, many of you will be aware, there's a large outbreak of avian influenza in Europe, North America, and more recently South America as well, with some spillover into human hosts. So again, having just an additional tool to be able to do genomic surveillance in the context of increasing amounts of self-testing and decentralization of testing, I think is going to be critical moving forward. And I've covered that. All right, so just to wrap up, I hope that I've, I guess, talked you through how outbreaks and pandemics can drive diagnostic innovations. I guess the challenge is how to maintain this in peacetime. And again, a sort of fiscal challenges, we're often get funded during pandemics, but that does drop off in peacetime. So how do we maintain that kind of innovative approaches and don't just go back to business as usual? I've mentioned this before, but there was a lot of duplication across the country. And from a laboratory perspective, I would just love to see some of the work joined up across the country. And I think this strong collaboration between the triple helix of academia, industry, and public sector, such as hospitals and reference laboratories, is really needed to really drive home the benefits of diagnostic innovations. So thanks very much for listening, and I'm very happy to take any questions. Thanks, Beck.
Thank you so much, Deb. That was incredible, and it was so amazing to, in a snapshot, those innovations in the COVID pandemic. I think many of us, we worked in had specific tasks, but I don't think anyone would have had a realisation of that series of innovations which were so impactful and also scaled up. So just congratulations on all that work, and I guess also all the innovations you've done regarding genotyping. Just really great to listen to. We've got a number of questions in the Q&A box, and just would encourage everyone to keep putting your questions in the Q&A box, but I'll just get started. Obviously, Deb, there's a lot of interest in the genotyping. I think you've touched on syphilis, and perhaps that's not something that has the same utility in real time, but you mentioned gonorrhea genotyping, and I know you're working in the HIV space as well. So Bridget's asked, could you give us some examples of public health interventions that could have occurred or can occur as the result of the genotyping epidemiological linking?
Yeah, that is a great question, and I don't think, as I mentioned, I'm not sure that we're there yet. But probably the best example of that, Bridget, is the Shigella work. So really showing that there was an issue with multi-drug and extensively drug-resistant Shigella amongst MSM. So what that enabled, I guess, was a tailored, not so much in some instances there was individual contact tracing, but a more kind of focused health promotion message around, if you have diarrhea, then with some, I guess, public health messages around that. If you have severe diarrhea, go to the hospital. If you and to clinicians as well, if you see a male coming in with diarrhea, make sure that you include a sexual history, which is not something that's intuitive to many clinicians, but also B, make sure that you send the sample to the laboratory for analysis so that you're not using antibiotics which won't be effective. So it was more, not so much at that individual level, but more at that broader kind of public health level. For gonorrhea, I mean, you can sort of see the possibilities. And certainly in the UK, for example, when there was an outbreak of XDR gonorrhea, there was an incident management team around that, and genomics informed that. So, okay, where is this strain tracking across the country? And so, yeah, a bit of a way to go, but I think the possibilities are there.
Thanks so much, Deb. There are some more questions about genotyping, so we'll continue on that pathway. So there's a question about genomics, and I guess it's, what does it tell us about undiagnosed cases? So how does genomic epidemiological analysis contribute to our understanding of undiagnosed cases?
Yeah, that's a good question. And so, first of all, the cases, it's a limitation of all genomic studies, really, the sampling frame. So, and it's important that that's acknowledged in studies. I'm always suspicious of studies that don't acknowledge, the fact that what is sequenced is quite a biased set of samples. So, they are, by their nature, samples that have come from people who've presented for testing, which immediately filters out, a lot of people. B, samples that have been successfully cultured and grown, which may, from a pathogen perspective, filter out some things. And C, also, a lot of the genomic data in the public domain may be because somebody has special interest in a particular AMR profile of a pathogen or a particular outbreak. So I think acknowledging and recognizing that upfront is helpful. How does it, I guess, so genomic epidemiological work is the tip of the iceberg. And I guess understanding that there may be many undiagnosed cases beyond that is important. But again, I think you can extrapolate that to any kind of notification data, right? You might look at it and go, okay, well, these are things that are notified, but there might be a bigger problem going on. Probably in terms of STIs, the most concerning thing is with gonorrhea. I think because at the moment, the work that's done on sequencing gonorrhea is on isolates that have been cultured. And so what that means is that these are isolates that have samples that have been taken, sent to a lab which can perform gonorrhea culture, and they are getting less and less. And at the moment, in Australia anyway, which is actually concerningly pretty good globally, that's only 20 to 30% of samples. And the rest is due to nucleic acid detection. So the vast majority of gonorrhea notifications are due to just PCR detection rather than culture. And for that whole, 60, 70, 80% of samples, we don't sequence those. So we only sequence the ones that are cultured. So again, that is a black hole in our surveillance, which is probably a reason to keep sequencing the other ones so that we get an idea of what's going on with the other 80%. I hope I've answered that.
You did, Deb, very comprehensively. Got lots more questions to come, so stay tuned, everyone. So diving into syphilis, this question is a little bit about, it's from Dave. Does the genotyping tell us anything about the contribution of the organism or its molecular changes over time? Have they contributed to the emergence of a super strain relative to other, I guess, environmental or behavioural factors? And I guess if I could add there, I guess important to consider susceptibility to certain antibiotics over time. But hand over to you, Deb, to respond to that.
Yeah, that's a really good question. And so probably the key thing that has been observed in Treponema pallidum, that noting that it is very difficult to do any phenotypic work on it to correlate what you find genomically with the phenotype of the pathogen, is macrolide resistance. So strains circulating, strains that people have had in their freezer and have been sequenced from before the late 20th century, generally macrolide susceptible. And we know that from a particular genotypic mutation. But almost all of the strains that are circulating globally now are macrolide resistant. Obviously macrolides are not the first line treatment for Treponema pallidum, but you could hypothesize that the increase in macrolide use, whether it's for respiratory infections, whether it's for STIs, the kind of collateral damage of that might be to drive macrolide resistance in other pathogens, such as Treponema pallidum. So we can see that genomically. We're not seeing at this stage any concerning mutations that may be associated with penicillin resistance. What is obviously of concern is that might happen, but also that we wouldn't be able to detect that in the lab in the same way that we would when we do antimicrobial susceptibility testing on, like E. coli, gonorrhea, Shigella, because we can't grow it. And so we'll be relying on clinical observations of treatment failure to look for penicillin resistance. But I still think that the pathogen per se is not responsible. Well, it is causal, but it's not. It's like there's a whole public health infrastructure breakdown, I think, that gets caused the syphilis epidemic in Australia and globally.
Thank you, Deb. I guess in terms of responses, it's taking on a slightly different tangent here, but I guess the work that you've presented is focuses on genomics for human health diseases, and also its implications for antimicrobial resistance. So thinking about One Health, which is animal, human and environmental surveillance, the question is about, can adding genomics to One Health surveillance, would that be excellent? No, that is excellent, but is this feasible in the medium term? So the feasibility of genomics in One Health surveillance. Big picture question.
Yeah. I mean, it's feasible, and other countries do it very well. And one of the exemplar countries for that is Denmark, actually. They've got a very good integrated One Health surveillance, which includes some genomic surveillance as well. So it's totally feasible. But it's the structural, political, economic, I guess, challenges to that are hopefully not insurmountable. And I guess it's pleasing to see that One Health features very strongly in the proposals for an Australian CDC. Is it feasible? And I mean, I think the pandemic showed that anything is possible. So absolutely feasible. And I mean, obviously, it would be, it would be amazing if that could happen here.
Thank you, Deb. We're going diving into the detail a little bit more now. So it relates to the question about undiagnosed cases, and Brendan's, I guess, interest to know if there's any statistical methods you're aware of that can ensure the data are more representative of the community. Is there any weighting methods or other ways to take that into consideration?
There are, and I usually defer to people who are much more clever than I am to do that. And I do work with a fantastic collaborator here called Sebastian Duchesne, he's an expert in this. And I know that there are models that can be used, I guess, to look at or to hypothesize, I guess, about the number of undiagnosed cases, if you like, as part of these genomic clusters. Quite honestly, I would be making it up, Brendan, if I told you exactly what they were. This is why I like to collaborate.
Thanks, Deb. We've got a couple of questions relating to your last study, which look very interesting, about genomics from the rapid antigen test. So Philip Keane noted, sorry, just go page down, didn't discuss it, but do you think genomics could make a particular contribution to wastewater sewage testing? For example, understanding where there are multiple lineages, outbreaks of a pathogen, which may inform control measures.
Yeah, that's a good question. This is such a hot topic at the moment, not just here in Australia, but globally as well. And we obviously saw, I think, the utility of. Don't forget this: the wastewater surveillance has been done for years for poliovirus, but not so much in a genomics context. And we saw the value of wastewater epidemiology, I think, during COVID. Again, looking at the emergence and spread of new variants, and that those wastewater data aligned very strongly with the clinical data. And in some settings, clinical genomics for SARS-CoV-2 is really, not used heavily now, but wastewater is used as a kind of aggregate way of looking at what's emerging. And certainly in Victoria, it was really used extensively. I guess from my thinking around this is that you might use wastewater, definitely a utility, but only if it's linked, I guess, to a very clear public health plan, and only if, I guess, there'd be actionable outcomes for it. So, detecting wild type or vaccine-derived poliovirus would be a good example of that, where you'd want to go in and do something. Similarly, if there was a very clear public health strategy around disease elimination, that might be something where that you would look for that particular pathogen, like measles, for example, or mpox or typhoid. I think they're the sorts of applications where you might want to think about wastewater epidemiology more broadly. It's a personal view, but rather than just, I mean, because you're going to find stuff in it, right? I mean, you know, but what are you going to, what are you going to do about it? How does it change public health, I think?
Thank you, Deb. Last few minutes, we've got from Francesca, who obviously knows genotyping very well. So we're interested to know a little bit more about the practicalities of the last study about the rapid antigen test. So she's interested to know if you could provide a little bit more information about where the cassettes were from. Was it from hospitals? How were they stored? And timeframes between sampling and extraction. I mean, for me, listening to that, it sounded like a really exciting opportunity, particularly for remote areas. And so, like Francesca, interested to know a little bit more about the practicalities. Thanks, Deb.
Yeah, I mean, very happy to share detailed protocols with you, Francesca, no problem. But broadly, for the SARS-CoV-2 rapid antigen test, the positive SARS-CoV-2 ones, we got the cassettes from the hospitals, testing in the emergency department, but also we bought some here, and we applied stored PCR positive samples to the cassette. So we had two different streams, so one collection from the ED and one where we had you know, devices, and we applied PCR samples. Were they stored in the fridge after testing? Yes, they were stored in the fridge in the ED after testing, and the time frame between sampling and extraction varied. And as I said, in a few instances, it was up to 8 days between sampling and extraction. We did see a follow-up in successful sequencing with longer duration, but we were still able to recover virus.
Thank you, Deb. It sounds like there might be lots of people interested in those protocols when they're available, like a really practical, important initiative. I might ask you the final question as the chair. Just in terms of COVID, and I know this question is asked of many people, but from your experience, particularly related to the diagnostic availability and evaluation and regulation, what have you learnt from that you think can be applied to STIs, HIV, or other infectious diseases in terms of the diagnostic pipeline?
That's a good question.
I know there's so much.
Yeah. I think honestly, the top line for me is early not technical, but it's more about engagement, early communication with the key people. And it wasn't clear to us in the beginning of the pandemic who they were. So a round table to discuss the regulatory aspects with the TGA, the scale-up aspects with the college, with PHLN, for example, economic aspects with industry and government funders, consumer involvement as well. So, you know, we had I think you were there, but we had a round table where we got all of those people in the room around rapid antigen test rollout, and that was incredibly useful. So early, open, transparent communication between all of the stakeholders, so you're not blindsided by one agency or one organization saying, oh, we don't want this to happen. That for me was not so much technical, but more around stakeholder engagement. Yeah.
Thank you so much, Deb, and that's a nice way to conclude the session today. So just a big thank you to my friend Deb for all her amazingness and sharing that with us today. I think we've all learned a lot from a presentation, the innovation, the genotyping, the diagnostics, the surveillance, working together. And so it was a really impressive presentation. So, and thank you everyone for joining us today. The presentation is recorded and it'll be shared subsequent in upcoming days. I'm not sure exactly when, but it will be circulated. So for those who weren't able to attend, you can forward it on, but it'll also be available on the Kirby website. So thank you very much, Deb, and thank you for everyone for joining today.
Thanks for having me. Thanks, Beck.